title is from preclinical to clinical. So it is not based on the clinical ECG, that also highlights on the preclinical data. Why? We all know that in research methodology, preclinical state is very much crucial, because for the discovery of a new drug molecule based on the preclinical data, CDSCO has approved this one based on only the preclinical data. So the clinical as well as the preclinical findings are very much essential for the discovery of a new drug molecule, right? So good morning, respected director sir, good morning HOD sir, my teachers, my colleagues and my dear students. So today I will discuss about the differences between human and rat ECG. So come on the BPharm first year level, anatomy of heart and some physiology of heart. Before starting the ECG, I want to discuss about the electrophysiology of heart. What are the function of heart? Do you know? Function of heart? Heart is a muscular organ that pumps the blood throughout the body, right? Right? And another one thing, the heart also produce the inherited and rhythmic electrical activity that is responsible for lifelong beating, right? So the major function of heart muscles are excitability, contractility and rhythmicity, right? Heart muscles contains several autorhythmic fibers. They generate electrical potential or action potential that are responsible for the signaling, electrical activity of the heart, right? So the heart has two major functions. One is mechanical and another one is electrical. What is ECG? ECG is a non-invasive technique, and that is also a composite recording of the action potential generated throughout the heart muscles, right? And heart muscle contains cardiomyocytes. You all know heart muscle contains the cardiomyocyte, right? And in the cardiomyocyte, at the membrane, cell membrane of the cardiomyocyte, various channels are there, ion channels are there, right? The voltage gated ion channels. But to understand the electrophysiology, we generally highlight on the sodium channel, potassium channel and calcium channel. Okay. And the inherited potential difference across the cell membrane, that is denoted as the action potential, you all know. Okay. So in the cardiomyocyte, they are arranged very close to each other. This is an implement. Okay. The cardiomyocytes cells are arranged like this. This one cell, this one cell, this one cell, and each cell membrane contains various ion channels, voltage gated ion channel. And inside the cell, that is negatively charged around minus 90 millivolt, that is known as resting membrane potential, right? And what happened at that time, the sodium channel gets activated, and we all know that intracellular sodium concentration is less than the extracellular region. That's why the sodium influx takes place. Right? Sodium comes inside the cell. Okay? So the intracellular sodium concentration is increased. That's why the voltage is neutralized and it reaches up to minus 70 millivolt threshold membrane potential. And after that, what happened? The potassium channel opened and simultaneously the calcium channel opened. Then what happened? The potassium efflux and calcium influx takes place inside the cell. That's why no voltage difference are there, and that is denoted in the plateau phase. If you see in the electrophysiology, you can understand. In the cardiomyocyte, sodium channel open. That's why sodium enters inside the cell. The voltage is increased, or the electrical activity, the minus 90 millivolt, that is neutralized, and that in that portion depolarization takes place. Okay, that is the phase 0. Okay, and after that the potassium channel gets opened. Ultimately what happened? Potassium efflux takes place. Right. So the repolarization takes place, and after that the calcium channel gets activated. At that time, potassium influx and calcium influx takes place. No voltage difference are there. That's why that is known as plateau phase. And after that, after few milliseconds, calcium channel closed and the potassium channel opened. The potassium efflux takes place. That's why the repolarization takes place. Okay, repolarization. Okay, have you understood? Okay, good. So go to the introduction. So ECG, as I told earlier, ECG is a composite recording of the action potential produced by the heart muscles or the entire heart autorhythmic fibers. And ECG is the graphical representation of the electrical activities. Right? So, as I told earlier, that cardiomyocytes are present like this, closely arranged. That's why when the first cardiomyocyte have the voltage difference or have generated the action potential, automatically in the junction of the cardiomyocyte, the potential difference is generated. That is denoted as the transmembrane ionic potential. Transmembrane ionic potential, right? So here is a video that is the electrical activities. As we know that the SA node, sinoarterial node, is known as natural pacemaker. It generates electrical signal, and when that is reaches in the AV node re-amplify it, and after that it passes to the bundle of His, and after bundle of His the Purkinje fiber, and the electrical signals are generated, and that is responsible for the contraction of the heart, right? So I can't pass it. So here there is the SA node. SA node generates impulses and that reaches in the AV node. It re-amplified and after that bundle of Lees and Purkinje fiber. Okay? Okay? Any query? Any query? Okay. Okay. So yesterday, Shomnath sir beautifully discussed about various parts of ECG, ECG interpretations, and ECG graph. First, I go to the general introduction of the ECG graph. That is the ECG graph. Okay. So that is the P wave. P wave, QRS complex, and another one is the T wave, right? P wave. P wave denotes the arterial depolarization. Arterial depolarization means sodium channel is open. Just imagine, sodium channel open, sodium ion enters inside the cell. Okay, arterial depolarization. So, in case of any arterial disease, rheumatic fever, coronary artery disease, in this case, the abnormality is shown in the P wave, right? Is any arterial abnormalities that is represented by the P wave. Is that clear? Okay, good. Then we come in the QRS complex. QRS complex. QRS complex denotes ventricular depolarization. Ventricular depolarization. Here one question arises from your side, I know that. Why ventricular depolarization or why not arterial repolarization? You know?
We want the question that side. We don't want you to just silent, please. While there is a question, we have to interact.
So this is a question. Sir provoked you. But did it come to your head? After arterial depolarization, why there is no repolarization? Why? How many arterial dysfunctionalities you know? Sir mentioned 2. Any more I want to hear from the audience. You know arterial dysfunctionalities? Due to cardiac rhythms?
Arterial fibrillation.
You don't heard about arterial fibrillation, arterial flutters? Heart? Which one?
Septal defects.
Sir mentioned coronary artery disease, where specifically artery part is there. Anything else you know? Achha. So the question is, why this question didn't arise, but what you think, what is your anticipations? Why after arterial depolarization? So you understand the mechanism of this action potential, is it clear? What you understood? What exactly you understood? What you exactly understood? We have our volunteer with the mouthpiece, so please I would be very happy to see you take very active participation and you must be eager to answer such questions. It is not a theoretical class, it's completely practical oriented teaching. So you have to have your practical knowledge. Anyone please? কে বলবেন হাত তোলো কোন ইয়ার তোমরা বিফার্ম কোন ইয়ার বিফার্ম which year maximum you are from which year third year So you completed your cardiovascular in pharmacology, right? CVS is done. You have the cardiac arrhythmia chapter also covered.
As well as maths, that is in the BPharm syllabus first year.
Yeah, BPharm only they are saying, that is BPharm, maximum BPharm here. So the question is, if you understood the total mechanism of the rising of from the resting potential from the minus 90 millivolt and then the action potential rising, depolarization, then there is the hyperpolarization or repolarization. If you understood the mechanism in between the plateau phase, the question is given to you that after arterial depolarization, after rising through the resting phase, when there is the depolarization, you give the tip of the P wave, right? So after that, why we are talking about ventricular depolarization but not arterial repolarization? That is what is the question. Thank you, sir. Am I correct? Okay. Yeah.
Anyone? Okay. So arterial repolarization takes place, but it is masked by the QRS complex. Okay? Always remember it. Another one thing, QRS complex. QRS complex is generally
Sir,
known.
I don't think they got this QRX masking, so you have to really explain this.
Okay. So the next part is the QRS complex. QRS complex generally denotes the ventricular depolarization. Okay. And the T wave that indicates ventricular repolarization, and sometimes U wave arises. What is U wave? That is the U wave. U wave generally denotes the repolarization of the Purkinje fiber or bundle of His. Okay. Repolarization of bundle of His. That, but sometimes it occurs. Even Somnath sir yesterday has discussed about the U wave. Okay. So any type of abnormalities in the ventricle that is denoted by the QRS complex, as well as in case of the ventricular tachycardia. Do you know that what is tachycardia and what is bradycardia? Increase than, more than? Yes, tachycardia, right?
No chorus answer. Anyone, please.
When pulse speed gets less than 60,
Heart rate.
yes, gets 60 beats less than 60 beats,
Yes.
then it's called bradycardia.
Bradycardia.
And after when it is done 100 beats, then it's called the tachycardia.
More than, right?
More than 100.
Exactly. Then that is denoted by the QRS. In case of the ventricular tachycardia, the abnormality is shown in the QRS complex. Is that clear? And there is several intervals. PR interval, RR interval. Okay. What is PR interval? That differs. PR interval. Timing between P wave and the peak R wave. PR interval. And PR interval, that is the time between the arterial and ventricular contraction, means arterial depolarization and ventricular depolarization, right? Understood? Okay. And another one is the ST segment. ST segment. S and T. T wave denotes ventricular repolarization. Very good. So ST segment that indicates ventricles are depolarized. That indicates actually whether the ventricle gets depolarized or not. Okay, repolarized or not. Okay. So that is the electrophysiology. As I told earlier, that in the cardiomyocyte, the membrane of the cardiomyocyte several ion channels, voltage gated ion channels are there. First, the sodium influx takes place, that is denoted as depolarization. Sodium ion enters from outside the cell to the inside to the cell, and there is a minor increase of the voltage, minus 92 and minus 92 and minus 70, and after that 0. Okay, and after that the initial repolarization takes place by the opening of the potassium channel. Okay, potassium efflux takes place. Potassium efflux, not influx. Remember that. The potassium efflux takes place. Okay, initial repolarization, that is the phase 1. Okay, after that, that is the plateau phase. In the plateau phase, calcium influx and potassium reflux takes place simultaneously. That's why no voltage difference are there. That is denoted as the plateau phase. Okay, and after that, the calcium channel is closed. Ultimately, what happened? Potassium comes out from the cell, potassium efflux takes place, and that is denoted as the repolarization, phase 3. Okay? Good. Acha. Please open the video. In the next slide. Yes.